Alright, welcome to my class. This video is about DNA replication during cell division. So when we say DNA replication, it's like making more copies of the DNA. It's just like you taking a document to a business center and then you do a photocopy. So the DNA can go through that same process of copying the information within its molecule. So during cell division, each daughter cell receives an exact copy of the genetic information from the mother cell. So the process of copying DNA is called DNA replication. So in your exam, I can ask you this particular question. When I ask you, what is the process of copying DNA called? The answer is DNA replication. And so in the daughter cell, one daughter strand is derived from the parent. So as you can see in this image, you can see that this color blue signify the double strand of the parent or mother cell that are entangled. You know, DNA is like a double helical structure twined together. And now something will happen. They will be separated. Each of them will become a template strand. That means a guiding pattern. And new strands will be made or will be formed. And so the strand in orange is the new strand. So when we say semi-conservative DNA replication, it means that each original strand serves as a template, and that template can also be called a mold or a guiding pattern. So it serves as a template for the synthesis of a new complementary strand. And so the person that came about this experiment is Meselson-Stahl in the year 1958. Bacteria were grown in a medium containing the heavy isotope of nitrogen, that is nitrogen 15, causing all the bacterial DNA to be labeled with heavy nitrogen, as you can see in this image. Then these cells were then allowed to divide in a medium containing the normal isotope of nitrogen. So in the first generation, all the DNA molecules were hybrid. That means half labeled with nitrogen 15 and the other half labeled with nitrogen 14. In the second generation, two types of DNA molecules appear in equal numbers. The hybrid, that is the half labeled, and the completely unlabeled, that is the nitrogen 14 only. So it was this experiment that confirmed DNA replication as a semi-conservative in vivo. In vivo means inside living organisms. So I can ask you in your exam the name of the experiment that confirmed that DNA replication is semi-conservative. So the next thing you want to look at is the base pairing rule. So the base pairing rule is always maintained, and this means that for every adenine there is a thymine, and for every thymine there is an adenine. So adenine is a base that you can find in the DNA structure. Both adenine and thymine, they always pair with each other. So they will say for every A there is a T, and for every G there is a C. So we call it at GC, just to remember. And so these complementary bases are joined by hydrogen bonds between the strands. As you can see in this image, we have two hydrogen bonds that always pair adenine and thymine, and we have three hydrogen bonds between guanine and cytosine. Then what are some of the steps of DNA replication? So these are the major steps of DNA replication: the origin of replication, the DNA replisomes, the DNA polymerase, and the fourth is the RNA primer. So those are the four things you need to know during DNA replication. So let's talk about the origin of replication, also called the ORI. Please take notes, ORI. DNA replication begins at a specific site. Sites. You know, when you say, I'm going to my farmland, I'm going to my site to construct my building or something. So that place, that position, that site where DNA replication begins or starts from, I can twist the English in your exam, is called the origin of replication, abbreviated as ORI. Although in mammals we have multiple origins of replication, and they are collectively called the ARS, which means the autonomous replicating sequences. So, what is the meaning of ARS, autonomous replicating sequences? These are the things you should be writing down in your notes. And I encourage you, please watch this video like up to 2 or 3 times before you write your exam. So now the DNA replications, number two. DNA replication requires the participation of over 20 enzymes and proteins, collectively called the DNA replicase system or replisome. This can be another potential question. So the DNA replicase system can also be called the replisome. And how many enzymes does this system have? It has 20 enzymes and proteins, over 20 enzymes and proteins. Then these are some of the key enzymes. The number one is the DNA helicases. I normally use Tokwe and Gerard and Helen. H for Helen, T for Tokwe, and G for Gerard or something. So the DNA helicases, they include the topoisomerases and the gyrases. They are the one that unwinds the DNA. So to unwind the DNA can mean two things. To break the hydrogen bond between the pairs of bases, one. Number two, it also means to unzip the double helix structure of the DNA. So the helicases is special because helicases can unzip the DNA in two directions. Now in DNA there are two directions. One direction is the strand runs from a 5' end to a 3' end, and the opposite direction is from the 3' end to the 5' end. So the third thing you need to take note of is the DNA polymerase. So the DNA polymerase is abbreviated as DNAP. So when you see DNAP, that means DNA polymerase. Then if you see DNAPS, it means DNA polymerases. That is the plural form. So these DNA polymerases, when we say polymer, a polymer is like a giant molecule that is formed from monomers. Mono means one, right? Di means two. So if I say monomer, I means 1 unit. But if I say dimer, I means 2 units. So if I say oligomer, that means I mean a few units. And if I say polymer, I mean many units. So when you hear polymer, it means that you are forming a giant molecule from the monomers. That means the single unit from the monomeric unit. So when we say polymerase, if you notice, all enzyme ends with ase, most enzymes. Let me not say all. And so when you say polymerase, DNA polymerase, that means is an enzyme that can create the DNA polymer. And how does it do that? By adding the monomers together. So what are the monomers of the DNA called? They are called nucleotides, and we're going to see that later. So what this DNAP does, or this DNA polymerase, is that it adds nucleosides together and make sure they form the giant molecule called the polymer, or the DNA itself is a polymer. So the DNA polymerase are enzymes that synthesize a new complementary DNA strand by adding nucleotides. You have to take note of the direction. It's not 3 to 5. It's not 1 to 2. It's not 1 to 1 million. It is 5 to 3 direction. This is the direction that the DNA polymer will always synthesize a new complementary strand. And like I said before, it must use a DNA as either a template, or you can call it a mold, or a guiding pattern. Now, DNA polymerases are different in bacteria and mammals. So in bacteria, bacteria is representing the prokaryotes, and the mammals are representing the eukaryotes. So in bacteria, there are three main DNA polymerases. So there's one we call the DNA polymerase 1, the Kornberg's enzyme, because it was discovered by Arthur Kornberg in the year 1959. Please take notes. So how does it function? It functions as a DNA repair enzyme, and it has both 3 to 5 prime and 5 to 3 prime exonuclease activity. Exonuclease means it can cut, or let me not say cut, unwind the DNA externally. So if I say endo, it's just like in biology when we have a exoskeleton and endoskeleton. So exo means external and then endo means internal. And so exonucleic activity means that it is cutting, like a molecular scissors, it's cutting the DNA externally. And so this convex enzyme can do that in both direction, from 3 to 5 prime end and from 5 to 3 prime end. Then one thing you have to also take note of is that the DNA polymerase 3, we normally write enzymes in Roman figures. You can see the Roman figure 3. The DNA polymerase 3 is the main replication enzyme in bacteria. This can be another question. What is the main replication enzyme in bacteria? It is the DNA polymerase 3. So now in mammals, we have 5 main DNA polymerases. We have 5, and these are their names, and this is what they do. Just read them, basically what they do, and their symbols. Oh, please also take note of their symbols, their name. So what I want you to take note of in this table is the name, the symbol, and the function of each polymerase in mammals. Don't confuse them with Cranberry and DNA polymerase 3, the ones we have treated earlier in bacteria. So another thing you need to take note of is that the mammalian DNA polymerase work at a rate of 100 nucleotides per second. They are not that fast, whereas the bacterial DNA polymerase work 10 times faster. So 10 times 100 is what? 1000 times. Then the RNA primer is the fourth thing you need to take note of in DNA replication. The RNA primers are required for DNA synthesis. They are primers, that means they are short oligonucleotides. They are just around 100 to 200 nucleotides long. They are RNA basically, and they are synthesized by these enzymes. The RNA primer is the name, but RNA primase, you see it's ending with ASA, is the name of the enzyme that will synthesize this primer. So this primer is just like it serves as a, like, I talked about the ORI, the origin of replication of a replication. So the foundation, just let's just call it the foundation. So the site is the origin of replication, but the foundation where other things are built on is the RNA primer. It is on this foundation that the DNA polymerase will be adding more nucleotides. So later, after the DNA has been constructed or formed, the DNA polymerase, the DNAP, is going to use its exonuclease activity to replace the RNA primer with DNA fragments. So now, what do we call the nucleotides? Like I told you earlier, DNA is a polymer, or we can call it a polynucleotide. It is made up of thousands, millions, billions of nucleotides that are joined together. So when we say a nucleotide now, one monomeric unit of a DNA or RNA molecule, it consists of 3 components: a nitrogenous base, a sugar, or a phosphate group. So this picture shows us a perfect difference. It highlights it for us to see between a nucleotide and a nucleoside. Nucleoside contains just a nitrogenous base and a sugar. The sugar could be a deoxyribose, that means it has no oxygen in it, or just a ribose sugar, the one that has oxygen in it. And so another key difference between RNA and DNA, I'm diverting a little bit, means that RNA has nucleotides made up of um, ribose sugars, but DNA has the oxoribose sugars. That's why it's called deoxyribonucleic acid. And so that's it basically. So a nucleotide consists of 3 components, whereas a nucleoside, tide 3, side 2. And so you need to know those 3 in the nucleotide and those 2 parts in the nucleotide. So I can say, in conclusion, I can say that the nucleotide is equals to the nucleoside plus a phosphate group. And I will be right in that sense. Okay. Remember, DNA is called a polynucleotide. In this video, we are going to look at the DNA replication process properly. But before that, I need to talk about the DNA structure. So the DNA is a molecule that is composed of two strands twisted around each other in a double helix shape. Now, each strand, as I've said earlier, consists of a sequence of four chemical bases represented by the letters A, G, C, and T. That is adenine, guanine, cytosine, and thymine. Please also take note of that. I may ask you. So the two strands are complementary. This means that wherever there is a T, there is A, and vice versa, as I've told you for G and C earlier. But each of those strands has two ends. One will normally run from a prime end, 5' end, to a 3' end, and the other strand will run in the opposite direction. That means from 3' end to a 5' end. And so the first step in DNA replication is the unwinding of the DNA, followed by the primer synthesis, the RNA primer synthesis, then the elongation of the new DNA strands. Then we have the last stage, which is the primer removal and ligation process. Okay, now let's talk about the unwinding of the DNA. So this unwinding of the DNA is also called unzipping of the DNA, and it is usually the first step in DNA replication. So it's the separation of the two DNA strands, and the process is called unzipping or unwinding. It's carried out by the enzyme helicase, and this enzyme will unwind the DNA and it forms a structure called the replication fork. So this is an example of what the replication fork is all about and how it looks like. Then after that, the separated strand, each of those two separated strands will serve as a template, as I said before, a mold or a guiding pattern to create a new strand of DNA. So this enzyme called the RNA primase, remember I said something about foundation. So it initiates the process by synthesizing a short segment of RNA known as the RNA primer. We already said that before. How many nucleotide long should it be? Remember, 100 to 200 nucleotide long. So this primer serves as the starting point for the construction of the new DNA strand. Remember, please. So that is where the origin of replication is along the strand. The site where the RNA primer will bind to will be called the origin of replication, or the ORI. And the RNA primase enzyme is responsible for building the RNA primer that will bind. So immediately this binding occurs, we go to the elongation of the new DNA strand. So an enzyme called DNA polymerase, that is the DNAP, will bind to the RNA primer and begins to add new nucleotides, new DNA nucleotides to form a complementary strand. So where if the parent had T, the new strand will have A. If the parent had, where the parent had a C, the new strand will have a G, like that. So that is the complementary, the base pairing rule. So the DNA polymerase can only add nucleotides in the 5 to 3 direction, like I told you to take note of earlier. So that particular new strand or daughter strand runs from a 5' to a 3' direction. So we will now have two daughter strands. We have the leading daughter strand and the lagging daughter strand. So leading, like the word signifies, leading means it's a flow, it's a continuous flow. But lagging means there is a break. So the strand where there are breaks or chunks are called lagging strand. But the strand that is continuous, that does not break, are called leading strand. So that leading strand is the new strand that is synthesized continuously in the 5 to 3' direction without interruptions, without any kind of break, without any kind of fragments. But the lagging strand is the strand that runs in the opposite direction, and it makes continuous synthesis impossible. It means it will be discontinuous. There will be breaks, there will be fragments. So the DNA polymerase synthesizes the lagging strand in small segments known as Okazaki fragments. Then after all the strands have been formed, the enzyme exonuclease will remove all the RNA primers from both the leading and lagging strands. And then DNA polymerase will fill in these gaps left by the RNA primers with DNA nucleotides or DNA fragments. Then an enzyme called DNA ligase, I'm going to ask you the function of this enzyme, seals the gaps between the Okazaki fragments and even those gaps that the new DNA fragments have filled in. So what ligase does, ligase is just like a joining enzymes. It joins all the DNA fragments in both strands together to make a completely new DNA structure that is semi-conservative. That means that that still has a one strand from the original parent's DNA. So mutation. When we say mutation, the first thing that should come to your mind is a change. So a mutation is defined as a change in the nucleotide sequence of DNA. It can be in a large scale, affecting large sections of a chromosome. The large scale is called the gross mutations, or it could be in a small scale called the subtle mutations, where it just merely involves changes in one or few nucleotides. So mutations may also arise spontaneously and abruptly, leading to the appearance of new traits. Statistically, it is estimated that one mutation occurs in every 1 million cell divisions. Normally, in every 1 million cell divisions, you have one mutation. So mutation can be classified into two examples. So we have the point mutation and the substitution mutation. So when we say a point mutation, there are mutations that involves a single nucleotide. This change have different effects. Silent mutation is an example. In silent mutation, no significant effect on the protein. Missense mutation changes one amino acid, possibly affecting the protein function. The nonsense mutation is also another example of point mutation. It creates a premature stop codon leading to incomplete proteins. Then the last type of point mutation is the frame shift mutation. This frame shift mutation can lead to a lot of dangers. It is caused by the insertion or deletion of a nucleotide, altering the gene's reading frame. So if it causes severe consequences by affecting the production of, it leads to the production of non-functional proteins. Then the second classification of mutation is the substitution mutation. In this type of mutation, like the name implies, substitution, one nucleotide is replaced with another. It can be classified as transition substitution, whereby a purine, that is an adenine or guanine, so purines are also called adenine and guanines, is replaced by another purine, or a pyrimidine, that is thymine or cytosine, is replaced by another pyrimidine. But a transverse substitution mutation happens when a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine. So that's all for now.